introducing Dr. Alicia Martin, who's from the Broad Institute. And Alicia got her PhD in genetics from Stanford, worked with Carlos Bustamante, who many of you know well. And she then did her postdoctoral training at the Broad and with Mark Daly. And throughout both her PhD and her postdoctoral fellowship, she was really interested in an area that is of very great interest to us, which is the rise in disparities and the potential for disparities that's going on with the bias in European ancestry GWAS. This is something that I think many of us saw coming down the road, but what Alicia did was sort of crystallize it in several very well argued papers in quantitative ways, and also I think was beginning to show the direction forward out of the quagmire that human genetics has worked itself into. So I will let her take over.
Thank you so much. I really appreciate that nice introduction. I'm going to go ahead and start sharing my screen. So you'll see my desktop at first, and then hopefully we'll get on the same page. Okay, so I think you should be, whoops. Well, I think you should be seeing, sorry, let me pause this really quick and make sure I've got the right set here. Apologies. Yep, sorry, wrong slide deck. Okay, now you should be seeing my, nope. You would think 2 years into this, I would have figured this out by now.
It's all good.
All right, now you should be seeing my screen.
Good.
Great, so I really appreciate that kind introduction. Seeing folks at UCSF is always a delight and a privilege. I think UCSF was one of the last places I was lucky enough to visit before the pandemic. So I enjoyed my visit in late October 2019. So it was wonderful to meet many of you, and I hope we get a chance to meet in person at some point in time again in the future. Okay, so just to kick off, I'm a population geneticist. As a population geneticist, I like to start by thinking through this evolutionary and historical lens. So I like to start at the beginning in Africa. So in genetics, that's where humans originated. As a species, we originated on the order of hundreds of thousands of years ago. We took a subset of that genetic variation out of Africa as our species migrated into the Middle East, into Eurasia, into Oceania, and subsequently into the Americas. So this human demographic history is really important to think about when we're considering how humans vary across the world. So one thing I think that's also important to emphasize when thinking about this framework is that there's more variation within than between populations. And this ancestry framework gives us this continuum. There's no discrete boundaries. Thinking about our DNA gives us sort of a continuum of complexity and genetic variation. Additionally, this human history over the course of the past 100,000 or so years informs a landscape of genetic variation across the world. This is really then important to think about when we're designing our big genetic studies.
Awesome. Do you mind if I eat? No.
Okay, so why genetics? Our genetic studies have just grown explosively over the course of the past decade and a half or so. So I think it's the most exciting time to be working in human genetics, and I think many others agree, because of how much our studies have grown explosively and about the biological insights that this has enabled. So around 2006 or so, when I was first starting to be interested in and training in genetics, our studies had on the order of hundreds to at very most thousands of individuals per study. And we're now routinely seeing that there are millions of individuals in many different disease consortia and different trait-related consortia. So that's really exciting. It's enabled us to learn a ton about human history, like I was alluding to, by putting together pieces from genetics as well as anthropology. But it's also enabled us to learn a ton about the biomedical basis of disease from the exponential growth that's taken place over the last decade or so. This also serves the genetics mission, which I see as providing unbiased biological hypotheses which then fuels downstream functional analyses and hopefully eventually therapeutic insights, which we're already starting to see very early benefits from. What we really, I think, need though, is to recognize the huge diversity problem. And it sounds like, I mean, UCSF folks have been on the ball with all of this for a long time, as have many others. We were really interested in this diversity problem. And one clear and critical issue is the fact that right now about 80+% of participants in our large scale genetic studies are of European descent. And this is vastly out of step with a global population where about 16% of the world's population is comprised of individuals with primarily European ancestry. So that's a huge problem. And it actually hasn't been getting any better for a long time. People have been pointing this out for over a decade. And I would say that the trend has not gone in a clear diversifying direction, which inhibits progress for everybody. So for example, diversifying progress peaked perhaps around 2014 or so, when it was still rather abysmal, and since then has stalled or perhaps slid in the wrong direction. So this is a really big problem, because if we have fewer genetic variants, we're then destined to find fewer genetic associations. And this is kind of what we're getting at when we have a Eurocentric study bias. So this has been a really pivotal issue for a very long time, and we've been working to correct these issues in genetics. But I think we've recently also started to recognize how much of an issue this lends itself to translation of genetic technologies. I think it's also important to recognize that this is a pivotal time as institutions sort of grapple with how ingrained issues of scientific racism are throughout science, but also more broadly in society. Okay, so with that as some backdrop, I'm going to talk through some lessons for genetic studies of diverse populations. We'll start with how critical diversity is for equitable translation of genetic studies. Then we'll move into the idea that multi-ethnic studies are uniquely informative of genetic and environmental determinants of health. Then I'll talk a little bit about where we go from here, both as a field and then some things that my lab is doing specifically. I should also mention my lab is only about a year old. I'll come back to the fact that we're definitely growing. So if folks are interested, please reach out. So onto this diversity being critical for equitable translation of genetic studies. The first place that we've started is with a polygenic score. So I imagine that many of you have heard of polygenic scores before. I'll just walk through some of the basic ideas here. So typically in a genome-wide association study, you have a cohort of patients or a cohort of people with some quantitative trait. And then often you're comparing this, if you have patients, then you're comparing this to non-patients or controls. You extract their DNA and you have this phenotype information that you're coupling with this genetic information. You scan across the genome and you identify associations between their genetic variants and their phenotypic outcome. So one thing that we've noticed is that across almost every complex disease that we're interested in, it's not just one variant that's contributing to your risk of disease. So we can't just pick up one gene and say, this is definitively whether you have a disease or don't have a disease. That happens in Mendelian disease. That's very uncommon for the majority of diseases and for the greatest health burdens in our society. Instead, what we're seeing is that there are teeny tiny effects scattered all across the genome. And to pick up these effects, we need huge numbers of individuals to identify these and to accurately quantify their effect. So this is why we're often seeing the need for actually millions of individuals in our genetic studies. So we can take this information that's scattered all across the genome to construct a polygenic risk score. So this uses summaries from your genome-wide association studies to basically aggregate a risk prediction from your individual's DNA. So basically you're taking effect sizes that you learned from GWAS, you're multiplying those by the genetic variants in your patient population, in a new patient population, you're summing those up across the genome, and basically that's your score. I should note that there's been a lot of conversation around these, which we'll talk a little bit about. But one, I think, area that we should be really cognizant of is how useful is a polygenic score at this point. So in this bottom right little plot of distributions, you can see this distribution of polygenic scores for many different individuals who have a disease versus many individuals who are healthy and do not have that disease. So you can see that there's a shift here. So individuals who have the disease tend to have a higher polygenic score. They have higher risk because they have more genetic variants that are predisposing to risk, and some combination of environmental factors potentially that's contributing to that risk. Whereas individuals who are healthy and lack this disease have lower risk, likely both from genetic and environmental influences. So they may have fewer genetic risk variants overall, and perhaps a lower environmental contribution as well. But notice that these distributions are really largely overlapping, and that it's only really in the extreme tails in which we're seeing really good ability to discriminate between cases versus controls. So right now, polygenic score is probably not going to be too meaningful if you are sort of in the kind of average part of the population, but only if you have, say, the extreme 1% or extreme 0.1% of risk from a genetic perspective will this be potentially really meaningful. That's not to say they're useless. So in preventative medicine, we use risk stratification tools all the time, and we couple these together with other risk factors. So for example, in the cardiovascular disease space, there's good clinical models that aggregate all sorts of different factors, including things like your LDL cholesterol, your family history information, whether you have hypertension, your BMI, all sorts of different factors. And you can imagine layering on a polygenic score as basically just another biomarker for how likely you are to have the disease. So a polygenic score has been talked about in a whole variety of different contexts. So the first is in a research setting. So in this scenario, we often think about like deeply phenotyped cohorts. So GWAS are really useful at aggregating sort of a general phenotype, but what happens when you want to learn more specific details about a patient's longitudinal trajectory? So I work in psychiatric disorders often, a whole broad spectrum of traits, but that I am based in the Stanley Center at the Broad Institute that focuses on psychiatric research. So one thing that we're interested in is not necessarily predicting whether you will or won't get schizophrenia. We could have conversations about whether that's useful. But if a patient has schizophrenia, how likely are they to be readmitted to a hospital? How likely are they to respond to antipsychotic medications, and so on and so forth. So this is like a context where we can imagine polygenic scores allowing us to probe along their genetic risk profile, how likely are they to respond to different interventions. Another scenario is in clinical trials. So this is a scenario where we are imagining identifying individuals at the highest risk based on their genetic profiles for having a given disease that a drug is targeting, let's say, or a behavioral intervention is targeting. In those scenarios, anything we can do to identify individuals who are at highest risk de-risks basically the clinical trial endeavor and is therefore likely to make these somewhat more cost efficient, given their insane cost as it is, that benefits everybody. The last couple of scenarios are in clinical trials and clinical use, and in embryo selection. People have talked about these things a lot. So in clinical use, I think the jury's still out on how useful polygenic scores will actually be, but this I think has also the most hype around it, especially in the context of, say, preventative medicine, for example. So one area that I think holds potentially the most promise is in an area like breast cancer, where you have relatively comprehensive clinical models, but they're still capturing a very tiny fraction of actual patients who manifest with the disease. A polygenic score has been more predictive of whether someone will have or develop breast cancer than these existing clinical models for about half a decade or so. So at least in European ancestry populations, that means that we've got a lot of room to go for non-European ancestry populations, but also that we have like a much more powerful tool than what we're currently using. The embryo selection case, we've talked about, I think it's a very dubious and problematic area. I'm not going to get into that too much today, but it's another area that's been a heated conversation, I guess, in the polygenic risk score space. Okay, so with that sort of background then, a core area and question that we're sort of interested in is how do ancestry study biases in genetics actually impact the generalizability of our knowledge? When I was coming from Stanford to Boston and was trying to think about how my work in population genetics could potentially bear on statistical genetics and how we think about complex disease and traits, this was the core question that was kind of guiding me throughout all of these research projects. So there's a few, I think, core considerations that we need to keep in mind for these. So the first is that we mostly expect causal variant effects to be shared across populations. So by that, I mean, if you could go and learn really definitively which genetic variants are causing disease in one population, and then you were to CRISPR that genetic variant in another individual, you should see their relative risk rise. Of course, that would be an incredibly unethical experiment, so we don't do that, but that's just what I mean by definition here, basically. The second consideration, though, is that allele frequencies, linkage disequilibrium, environment, natural selection, and other complicated factors all influence how we aggregate genetic variants across the genome due to these population history differences. And therefore, we expect our population history to really come into effect again when we're thinking about generalizability of genetic technologies, particularly when we keep studying sort of a biased subset of the human population. Okay, so we first started on this and published a paper in 2017 that was looking at human demographic history and was coupling basically population genetic simulations with statistical genetics complex disease models. So in this paper, we're trying to address exactly that question that I stated on the previous slide, but without the massive accessibility and availability of data and biobanks that we have today. So we did both some simulation-based analyses as well as some empirical analyses based on 1,000 genomes data, for example, and just empirical observations compared to observation. So I'm not going to go through all of the analyses here, but I'm just going to give you some of the highlights and key takeaways. The first was that genetic prediction accuracy decays with increasing genetic divergence between our discovery set and our target cohort. So what that basically means is if we're doing a GWAS over and over again in European ancestry populations, but we're trying to predict genetic risk and the whole breadth of human diversity, the more genetically distant those populations are, basically the further away you get from European ancestry discovery populations, the worse your genetic prediction accuracy is going to be. The second thing that we learned in this study is that polygenic scores differ across populations fairly arbitrarily, and these biases are not necessarily meaningful. So there's a figure I like to show here from the paper that looks at height and variation across 1,000 genomes populations. So in this scenario, we use data from the aptly named Giant Consortium to construct polygenic scores in the 1,000 genomes project dataset. And what we saw is that we predicted that European populations would be the tallest, followed by South Asian and admixed American populations. We predicted East Asian populations to be the shortest and African ancestry populations to be the second shortest. These are not trivial differences. So there's like 5 standard deviations of difference between the African distribution here and the European distribution. So if we're to try to peg these, for example, with real numbers, let's say the average European ancestry man is like 5 feet, 8 or 9 inches tall with a standard deviation of 2 or 3 inches. 5 standard deviations of difference means that the average African ancestry man we would predict would be somewhere under 5 feet tall, which is clearly very far out of step with reality. And these biases do not necessarily track with reality, and in fact are often caused by residual stratification, confounding, other complicated factors that we can't always account for easily in our genome-wide association studies or even understand right away. The last, yeah,
sorry,
go ahead.
just a question. So I think this is an interesting kind of question, is what part of these sort of differences are kind of, uninformative, meaningless, versus what part of those differences are actually meaningful. And I don't know if you're thinking about that kind of question. In other words, like if there's a disease that's more common in Europeans, like type 1 diabetes or whatever, and you generate a PRS and it sort of does move over to the right, or disease that's more common in African Americans, say prostate cancer. And people have done this actually in journals and said the PRS well shifts over to the right. But your paper clearly shows that drift could not be so distinguishing the meaningful versus meaningless kind of drift in the PRS across populations seems like an important area.
Right now I'm of the opinion that we are not at a state where we can draw conclusions about prevalence differences based on polygenic scores. I think we're kind of hopelessly confounded in that area. We don't understand well enough what's going on when aggregating genome wide to be able to, make sense of these prevalence differences. I think we've got much better other tools out there in epidemiology space to be able to make sense of that. And I'm not sure genetics is the answer there.
Okay. Sounds good. Okay. So you would recommend sort of like mean centering the PRSs at the very least for and making them kind of specific based on population incidents and so on, things like that.
Yeah. So posting these kinds of examples is something I've seen in a lot of following up on kind of this type of work. Others in sort of semi-pseudoscientist kind of journals have published these kinds of things, talking about all sorts of things like educational attainment and IQ and stuff. And I don't think there's any, doesn't hold any water. But if that doesn't hold any water, then we also can't expect more biomedical literature to hold any water when it comes to things like, as you're mentioning, like prostate cancer differences. Yes, there are clear prevalence differences, and some of those might be due to genetics, but a polygenic score is not going to tell us that. Maybe some individual loci that are much more common and that we can really grapple with will help us understand that.
Okay.
Yeah.
Thank you.
Sure. So the last kind of point here was when doing our population genetic simulations and sort of modeling complex traits, we were able to observe these kinds of differences with neutral human evolution alone without invoking any arguments of natural selection. So for example, there's been a lot of literature and even much more recent literature arguing about whether height has undergone polygenic adaptation. And I think the jury's kind of still out on the extent to which polygenic adaptation has potentially occurred or not occurred in this scenario. But we were able to show evidence of these kinds of biases without invoking any natural selection in our models. Okay, so that was one of our earliest studies kind of looking at this phenomenon. We followed up with this in a large scale UK biobank sort of study and analysis. So UK Biobank is a phenomenal resource of about 500,000 individuals all in the UK, where they've sort of systematically phenotyped individuals in a very consistent manner across the country. And so but of course, based in the UK, that means about 96% of participants in the study are of European descent and have European ancestry. So we then wanted to take that 96% basically, run GWAS, and then in a holdout set, assess how well we're predicting in other populations, in European ancestry populations, as well as populations who derive their ancestry primarily from other continents or from different groups. And so what we see here when we try to do this is what we could kind of expect to happen in the GWAS field generally, since primarily what we're seeing are big studies of European descent and pretty small studies of other ancestry groups where people are interested in applying polygenic scores. So when we did this, what we saw were really massive disparities in accuracy for polygenic scores. So for example, if we pegged our relative accuracy for 17 different traits in Europeans at 1, and then asked relative to that, how well are we doing in these other populations, we saw huge differences in accuracy. So for example, we were predicting in European ancestry populations about twice as accurately as we were in East Asian ancestry populations, and about 4 to 5 times as accurately as we were in African descent populations. Notably, I'm excited about the prospect of polygenic scores, since I can't think of a single lab test that's so predictive of such a broad spectrum of diseases for a relatively low cost of running a GWAS array. But this is what I see as the biggest challenge to implementation of genetics and precision medicine. Right now, we're stuck until we sort of fix this issue, I think. This is especially problematic because other areas of clinical medicine, I think, have not found that just as a function of who your ancestors are, you're seeing that tests are working so differentially. I think people would be really up in arms if they realized that, for example, like an LDL test was working so much better in one population than another, and that because of that, you get different quality of treatment. So this is kind of like a unique problem, I think, to genetics, but is something that we can overcome simply by choosing to study the more diverse set of populations. One thing that I think is also worth mentioning, I pointed out at the beginning that our reflection of our genetic studies is not at all capturing global diversity, but it's not even capturing domestic diversity, even though the US is the biggest funder of biomedical research, I think, kind of out there. Beyond that, then what it sort of is it reflective of? It's clearly very reflective of the investigators doing the biggest research out there. So in order to sort of broaden our diversity and broaden, I think those representatives in those participants in our genome-wide association studies, the only way we're going to, I think, be able to make headway is to also train a reflective body of researchers who we would like to see in these genetic studies. So sort of a tangent there. Okay, so we know that polygenic scores decay in accuracy just as a function of genetic distance from our discovery cohort and our target cohort. But why? There's a few key reasons here. One of the first is that our genome-wide association studies are best powered to discover common variants. So that means that we have sort of an ascertainment bias towards variants that are more common in European ancestry populations than elsewhere by studying European ancestry populations. And because they're more common in European populations than elsewhere, we have less ability to potentially predict phenotype or explain phenotypic variation in non-European ancestry populations. So we took a tally in the GWAS catalog and saw sort of a reflection of exactly what we expected. So for example, we see genome-wide significant hits popping up for all sorts of different diseases and traits that are more common in European ancestry populations and less common elsewhere. Another big driver of this predictable basis comes from linkage disequilibrium differences across populations. So LD is basically the correlation structure between pairs of SNPs along the genome. We usually see pretty consistent patterns of LD across populations. So for example, in this plot on the bottom right, I'm showing you a regional plot of LD. So the closer you are to a diagonal, the closer you are to looking at SNPs that are physically close to each other in genome space. The more off diagonal you get, the further apart two pairs of SNPs are. And so I'm showing you in the bottom left part here, a European ancestry population, and the top right part, an East Asian ancestry population. The darker blue indicates more correlation and the lighter blue indicates less correlation. So from this plot, you can see that most of the time we have pretty symmetrical LD structure and pretty symmetrical LD generally, but there are some exceptions. So here where this arrow is pointing, for example, you can see that there's a region of extended LD in East Asian ancestry populations that's not present in European ancestry populations. Telling you all about LD, but why does that really matter? So if we have a causal variant in one population and we try to transfer that to another population, but our LD, the basically taggability of what we're finding in GWAS is different between our causal variant and what we're actually including in our model, then we're by definition going to find a different prediction power and probably lower prediction accuracy as we go to generalize and transfer this across different populations. And then the last sort of set of issues is something we don't, I think, have a good grasp on right now, which is that there's really clear environmental, selection, and other complicated differences pertaining to how we might predict polygenic scores across populations. So for example, we've looked at a couple of populations or a couple of cohorts, one based in the UK and one based in rural Uganda. Based in rural Uganda, we know that malnutrition rates are much higher, for example, than in the UK, including with individuals with matched ancestry. How does that impact our phenotyping and impact genetic influences over that phenotyping is something that we've been trying to get a little bit of a grasp on, and that really, I think, impacts this sort of like latest point. Okay, so sort of transitioning from that topic, the next general area for lessons for genetic studies in diverse populations is that these multi-ethnic and multi-ancestry studies are uniquely informative of genetic and environmental determinants of health. So we've touched on this a little bit by comparing, for example, data from the UK Biobank versus data from the Biobank Japan. Before getting into that, I just wanted to give you a little bit of background about these two cohorts. You may also already be very familiar with them, so sorry if this is overview, but the UK Biobank, as many of us have used and are aware of, this is a general population. It was enrolled through a volunteer-based sort of criteria. So people who are choosing to volunteer tend to be healthier, wealthier, and have higher education than average Brits, for example. The Biobank Japan ascertained on 47 different diseases. This cohort went through hospital-based recruitment. So what that ends up meaning is that the participants in Biobank Japan tend to be sicker on average than your typical Japanese person. So I teamed up with a really talented grad student, Masa Kanai, to evaluate the promise of diversifying efforts in genomics. So our goal was to kind of see how symmetric and comparable our polygenic scores would be if we set things up as symmetrically as we could possibly imagine. So for example, we ran equal sized GWAS for 17 different quantitative traits and 5 different diseases in these two cohorts. So this meant we had, just to match sample sizes, anywhere between 80,000 and 150,000 individuals in our study. And then we computed within ancestry polygenic score accuracy as well as across population accuracy. So this provided what we thought of as sort of an ideal setup to identify whether there was symmetric and comparable polygenic score accuracy. So if we're using European ancestry populations to predict European traits, are we performing comparably to how we do in East Asian ancestries when our discovery cohort is from an East Asian discovery cohort? So I'm not going to take you through all of the results, but some key takeaways here are that our ancestry matched results were always performing better. So predicting European traits, we did a better job with European training data and vice versa for the Japanese data. But our largest meta-analysis is actually always performing best. So meta-analyzing and building the largest sample size we could from European ancestry plus East Asian ancestry traits was performing the best. The second and third points are pretty related, actually. So our heritability differed by cohort. Heritability is not a fixed innate entity. This sort of depends on your data and your context. So one thing that we learned is that we're doing a better job, and our heritability tends to be a bit higher for quantitative traits in the UK Biobank when we're assessing natural variation across the population. But we're doing a better job with phenotyping with higher heritability for disease traits when looking at the sort of disease ascertained cohort with Biobank Japan, for example. So these also sort of made sense and lended to the fact that we did not see a symmetric prediction accuracy necessarily. Okay, so that's sort of just like a jumping off point, but where do we go from here? So I'm really excited. My lab is like about a year old. We're just starting to kind of get some groundwork laid. So I'm just going to talk to you about some areas that my lab is moving in and that I think we're moving in generally as a field to facilitate more diverse population genetic studies. So this is what I think is kind of a roadmap for the group towards facilitating diverse pop gen studies, which is, I think, critical for health equity, especially for translation of genomic technologies. So we have some really big opportunities in genomics, I think. We have the opportunity, for example, because of our Eurocentric sort of study biases, to identify novel genetic associations that are more common in underrepresented populations. This is sort of low-hanging fruit that's just waiting to be discovered. We also have the opportunities, I think, to pinpoint causal variants through fine mapping by leveraging differences in population history and differences, therefore, in genetic correlation structure. Those multi-ancestry studies are the best for fine mapping at pinpointing causal variants, which then improves our generalizability of our fine mapping or of our polygenic score analyses, for example. We have a lot of opportunities for functional follow-up then by pinpointing these causal variants and testing our findings in biological models and in animal models, for example. These last two areas are the ones that my lab is pushing most in. So in genetic and environmental determinants of health, modeling these together And then, of course, looking at genetic prediction accuracy, sort of evening the playing field for genetic translation of these sorts of polygenic scores and other technologies, especially for underrepresented populations. So there's a bunch of big things in the way, I think, of us meeting these opportunities. So the first is in our statistical methods. Right now, we mostly assume some degree of homogeneity in our models, and our sort of study designs are often not set up to manage that degree of heterogeneity that we really want to be studying optimally. We also need better data and community resources. So right now in genomics, most of our data sets and community resources have really been geared towards European ancestry populations, and that's pretty ingrained. It's been happening for the past, it's been happening since the dawn of genomics, really. So we need to make better use of our existing data. We need to collate across existing data boundaries where data is typically not shared. And then, of course, we also need to grow to address the gaps where we just don't have good data at the moment. And then we need to develop concomitant resources to support all of those activities, particularly for diverse populations. And then, as I started to allude to, I think we also need to improve research capacity, because who we're doing research on is really reflected by who is doing the research. So I think in order to diversify our genomics, we really need to ensure that the next generation of researchers can lead in their home communities and in their home countries. Okay, so starting with making better use of the data we already have, we've spearheaded this effort, the Pan-UK Biobank project, which we're hoping to wrap up in the next few months, but we've already last year released a whole lot of genome-wide association study data in case you'd like to make use of this. So I mentioned that the UK Biobank is 96% of individuals of European descent. But that still means that there's tens of thousands of individuals of non-European ancestry groups, and there's like thousands of traits that have been cataloged by the UK Biobank. So in many cases, these are the largest cohorts of non-European ancestry descent for a lot of these different traits, but they've just been routinely kind of left out of UK Biobank analyses. So we just wanted to make it routine and easy to use for people to be able to just gather the latest multi-ancestry analysis from UK Biobank. So we've released a ton of summary statistics, and this has been a huge team effort for these thousands of different GWAS, basically. So we released 12 terabytes worth of summary statistics for 16,000 GWAS of 7,000 different phenotypes across 6 continental ancestry groups. You can learn all about that at the Pan-UK Biobank project. You can download as much data as you would like, and we've set up some tools for querying this massive scale of data. We also released some frequently asked questions, basically describing the project, what you can say from this data, what you can't say about this data. For example, thinking about prevalence differences from polygenic scores and where we kind of fall on that, and what good uses potentially of the data are for better understanding where gaps might be. Compared to previous sort of UK Biobank wide scans, we've also improved in methodology by making use of linear mixed models instead of fixed effects models with PCs. So here we're also modeling relatedness structure among individuals, and then better population genetic sort of relationships among pairs of individuals. We've analyzed a broader set of phenotypes, including phe codes, so aggregations of electronic health record codes, prescription data, COVID-19 case control status, basically, and levels of severity of COVID-19 response. And then we have many more analyses underway. Things that we're super interested in that we spent, I think, the most time on in the last year have been sort of related to heritability analyses, which has given us a really good grasp on which traits are really not great to be analyzing or which ones are going to be problematic. Polygenic score analyses, we're really interested in this idea of, like, once you've done the broader genetic study, how much does that actually improve your ability to predict across populations, even just by including, sort of tens of thousands of individuals of non-European ancestry descent? Are you actually getting something out of that in terms of prediction accuracy? And that's likely going to be genetic architecture dependent and phenotype dependent. Another effort that we are really involved in and really excited by is the Global Biobank Meta-Analysis Initiative. So this initiative is designed basically to move across existing collaborative boundaries. There have been many disease consortia that have taken this sort of effort on sort of for one disease at a time. We've kind of brought together folks who have spearheaded efforts at many of these different biobanks to bring this together for several different phenotypes. So as of September, we had over 2.6 million samples genotyped. You can see where all of these different biobanks are coming from. There's still some really big gaps in where biobanks are represented, also kind of where biobanks actually exist. But this does span sort of a broader spectrum than previously, I think, for large scale, just biobank analyses. So Wei Zhu is a postdoc who has just done a phenomenal effort kind of bringing all of that data together. But moving this forward, we've also been kind of focused on a few different disease areas. So one disease area that we've been especially interested in studying is in asthma. So we've been especially interested in asthma for a few reasons. One, I don't think I probably need to tell you, because we've got like a leading asthma geneticist sort of among your colleagues, and Esteban, for example. But of course, we recognize that there are huge prevalence differences in asthma across different groups. This isn't necessarily tied to just straight ancestry differences. So of course, there's some genetic effects that are clearly underlying asthma risk overall, but also there's huge environmental differences that are likely also affecting these disparities. So by aggregating data from 18 different biobanks, we've accumulated about 150,000 cases and 1.6 million controls to study asthma. And for asthma, this can either be doctor diagnosed or self-reported, depending on which biobank we're looking at. Looking across all of these biobanks, we're sort of vastly increasing sample size compared to what's been done previously. We've identified 180 loci in this big genetic study, about 49 of which are actually novel. Despite enormous differences in prevalence, you can see the prevalence differences ranging on the left side here and the sort of sample size on the right side of this plot. We've got prevalence differences or prevalences ranging from anywhere between like 2 and 3% versus up to like 20 to 25%. So really like an order of magnitude difference in what's being reported across different biobanks. Despite this enormous difference in what's being reported as a prevalence difference, we are seeing remarkable consistency in genetic effect sizes. So that indicates that no matter where we're studying asthma, we're still picking up relatively consistent biological pathways and genetic signatures of asthma. So that's promising. We've vastly improved, I think by about twofold or so, how much of the phenotypic variation we can explain using polygenic scores. So we can use polygenic scores now as a better sort of biomarker to try to dig deeper into some of these disparities and try to understand these. We also find high genetic correlation with some other diseases and disorders. So for example, COPD, chronic obstructive pulmonary disease, is one where we've kind of dove into this pretty deeply. But we also see pretty different etiologies kind of correlating here. So whereas asthma is pretty highly genetically correlated with COPD, other biological pathways that are enriched seem to all kind of relate to immune function generally. Whereas for COPD, we're seeing a lot of, for example, nicotinic receptors, and we know that it's a much later onset disease, suggesting that there's, of course, differences in epidemiology there. Through the Global Biobank Meta-Analysis Initiative, we've looked at 14 plus, or I think 14 different phenotypes so far. And we've looked at polygenic scores across this whole spectrum of different diseases and disorders. I should mention that we chose these different diseases to focus on as a consortium and a collaborative sort of effort across many, many different labs and groups, because they've typically been somewhat left out in disease-focused consortia. So, you know, there's many different diseases that are represented here that we don't often see in just like every routine GWAS analysis. That also means that we've been able to make pretty decent progress in improving polygenic score analyses. So Ying is a postdoc in my group, has been leading a lot of sort of best practices kind of analyses for polygenic scores, looking across different ancestry groups, looking across different traits with different genetic architectures, traits with different levels of prevalence across the world and with different heritabilities. So she's been trying to develop these sorts of best practices and computational pipelines to facilitate polygenic score analyses kind of more generally. We're also developing really big, large-scale resources. I mostly just wanted to point here to the HGDP and 1,000 genomes resource that we've jointly called, we've harmonized, and we've released this out to the world. So if you want to make use of this as like a reasonable reference panel that's more diverse than previous releases of 1,000 genomes, that's available here on the Nomad website, for example. So I'm just going to kind of skip over that since I'm running a little short on time. But other areas where we're like really excited and new investments in genomics generally are particularly focused in Africa. So since my PhD, I've been very interested in African genomics. Humans originated there and have the greatest genetic variation and often cultural variation. So I think this is a very good place to do our genetic studies. And in a vacuum, in fact, I think if we were to start anywhere, Africa as a continent would make the most sense. But due to logistical reasons, of course, that's not traditionally been the case. So one massive study that we are on track with is the Neuropsychiatric Genetics of African Population study. This is a study of 40,000 African participants. It's a case control study for psychosis spanning Ethiopia, Uganda, Kenya, and South Africa. We're taking on a sequencing approach. So we're doing low coverage sequencing, like glucosome exome analysis in there, to develop a big resource of whole genome sequencing data for the purposes of studying psychosis and learning about the genetic basis of schizophrenia and improving generalizability of our genetic studies and identifying hopefully new genetic risk factors for psychosis. There's a huge capacity building effort that's gone alongside this. So I'll tell you a little bit more about Ginger. So Ginger is a training program that consists of 3 big components. So the first is workshops. So these are kind of the easiest, lowest hanging fruit, where we just basically hosted research fellows in Boston and in London. But really the bread and butter of the program comes from online weekly classrooms. So this was kind of like before pandemic, we had already kind of implemented this sort of Zoom culture for research fellows spanning these different institutions in Eastern and Southern Africa. And then we also have research fellows as part of this training program that do on-site training. So they basically serve as TAs to tailor and develop curriculum for their institution so that they're not the only researchers at their institution who are well-trained in bioinformatics and genetics and statistics and so on and so forth. So they help develop a sort of cohort around them through these on-site trainings. We've had 17 research fellows go through a 3-year training program that all graduated in 2020. And there's a re-up of this program that's a 5-year program with a smaller number. So there's, I think, 8 research fellows that are going through this. And I've been totally delighted to take part in many aspects of this training program. I would mention, I think this is a really critical element of the research that sort of is needed to ensure that there's not just safari research, so that researchers are not just sending their samples to the broad, never to be heard from again. There's really a deep partnership that happens by doing this training. These are the researchers who are going to be leading much of the research that's coming out of this activity. So as part of this activity, I got to work closely and mentor several of the Ginger Fellows. So I worked closely, for example, with Nastasha, Lorato, and Alan, who you can see here. We're a group of not very tall humans minus Alan. So you can also see some of the work that we've done, basically following up from our previous work, looking at polygenic score accuracy across different populations. So we recapitulated what I had seen earlier across continental ancestry groups, but we also went in more deeply into African ancestry groups and saw that the differences in terms of how accurately we're predicting across different African ancestry groups, if you're just to take sort of fold changes in accuracies here, are basically as large as what you're seeing across different continental ancestry groups. So we have a ton to learn by studying underrepresented parts of Africa in our genetic studies as well. I'm going to skip through some of the kind of nitty gritty, but I just wanted to point out that.
Sorry, can I just ask a quick question about that previous slide? Because I think it's an important observation. So it looks like it's sort of, if I understand correctly, on the right panel there, it's sort of accuracy fades from Ethiopia as you get either farther west or south in Africa.
Yeah, exactly.
And So do you think it's just because Ethiopia is closest to this sort of to the bottleneck out of Africa?
Yeah, Ethiopia has gone through more, there's been back to Africa migrations that have occurred over the past couple thousand years, 3,000 years or so. And it's, people have migrated out of Africa back into Africa, and that's happened like through consistent waves of migration. So I think that's due to the fact that like the genetic divergence is lowest between these well-studied populations. For Ethiopia, in the FST, for example, or genetic divergence is highest among these population groups for Southern African groups. Right.
But even east, like it's sort of a little better than south, just because of sort of that closer to the bottleneck.
Yeah, I think so. And one sort of following up on that point, for East Africa, for East African ancestry individuals in the UK, we were able to compare to this separate cohort that's based in Uganda, and we saw poorer performance when we predicted across cohorts, but still the ancestry matched groups. Kind of coming back to what I was mentioning before, there's clear differences that are not just genetic, also relating to likely polygenic score differences that are being predicted here. So there's a lot of fun things to dive into. Larato's been spearheading this effort and is super talented. I'm delighted that she stuck around to do a co-advised postdoc with me, following her PhD and following her training through this program. It's been really wonderful. I'm just going to skip forward and say there's a few things that I'm really excited about. We're putting together not just genetic risk profiles and the simplest polygenic scores, but also socio-environmental risk models. So from the genetic side, we're moving beyond single trait, single ancestry analyses. We're doing multi-trait, multi-ancestry, polygenic score analyses, but also incorporating experimental data, functional information, to find where in the genome we're enriched for heritability, or we're sort of weighting those higher in our polygenic score analyses. On the environmental side, we're modeling the quote environment in a more systematic manner by also doing sort of factor analysis across the UK Biobank to combine risk factors there to overall model disease risk. Happy to get into that a little bit more. Please feel free to follow up, but just in interest of time, I'm just going to skip to sort of next steps here. So polygenic scores are really exciting, but because of our Eurocentric study biases, I think right now we're on track to worsen health disparities with genetic technologies due to these massive study biases. So to rectify that issue, I think we need more diverse GWAS, new methods, resources, and research capacity, which is going to be, of course, like a field-wide effort to address these major issues. And some of these topics are clearly quite culturally sensitive. So I've been really pleased, I guess, with sort of wider scale dialogues that have been happening beyond just our research papers, but have been happening out in society about genetics and about why our Eurocentric study bias is problematic. So I've left my contact information here. It's been a huge team effort. So, thank you to all of those who have contributed. And please feel free to ask any questions. Happy to take any if we've got some time.
I have a good question. Hi, Alicia.
Hi.
Thank you so much for this presentation. You've done a lot of work. So my name is Kimberly. I'm a postdoc at UCSF, and I'm also from Trinidad and Tobago. So this is of particularly interest to me, as I have an NGO in Trinidad, and we've been trying to set up the infrastructure to build out a biobank that's applicable towards the Caribbean region. And that has been the amount of challenges that we've had to go through, that is a whole other problem. But my main question for you is whether or not you've done any analysis in populations of mixed ancestry, and where they fall in their risk scores in terms of accuracy. Because, for example, in Trinidad, what we see is that persons of mixed ancestry, sort of like the incidence of breast cancer would lie somewhere between, the two mixes, whatever the two mixes are. Mm-hmm. So I was wondering if you had any data on that.
Yeah, I'm working closely with Elizabeth Atkinson, who's a colleague who's developed basically local ancestry-based approaches for conducting GWAS. And then we're also following up on that to look, for example, at polygenic scores. But exactly as you say, there's a lot of different ways that you can think about mixed ancestry populations and about how we would model the genetic basis of that. So we're going down this kind of like local ancestry path to sort of try to understand at finer scale resolution across the genome what's contributing. But also, I think it's really nice to have recently admixed populations to be able to sort of in a controlled way where the environmental exposures are somewhat, or they're the same for that individual across the entire genome, right? Understand and tease that apart. But I think we would be remiss not to recognize also that the cultural factors that impact individuals of mixed ancestry are also going to fall along some line. So there's a lot of complications and a lot of interesting work going on in that space. Happy to connect more and talk more about that if you'd like.
All right. Yeah, perfect. Thank you, Alicia.
Yeah.
Other questions? I think, Naren, I don't know if you're still on, but you had a question earlier.
Yeah, so environment and non-genetic risk factors and G by E, how much are those impacting height differences? I think right now, the strongest signals that we have are of ancestry-based differences. So basically genetic differences. We can explain a huge amount of the difference in how accurately we're predicting populations just simply as a function of FST. And basically our analyses show like a linear decline with FST. It's highly correlated. In terms of how much G by E is influencing height across the genome, there may be specific regions of the genome that are showing these sort of risk factors. But for height, I haven't seen too many that have shown G by E effects in humans. There may be some, but I'm not super familiar with that literature.
Thanks very much. I think I asked that question quite early in the talk, and then I really enjoyed hearing about the heritability estimates and factoring that all in. So thank you. It was great.
Awesome. Thank you. Nice to meet you. And yeah, please reach out if you have other questions or want to chat anymore.
I had one other question actually, which I'm just wondering, it came from kind of the cancer literature, and just wondering kind of what your thought is. So in cancer, one of the big disparities, or I don't know if you want to call it a disparity exactly, but one of the big differences that we see in the somatic spaces are EGFR mutations in lung cancer. They're about like 40 or 50% of tumors from lung cancer patients in East Asian ancestry have an EGFR mutation, which is a driver and a treatable target of disease. And it's only about 10% in European ancestry, and I think it's around the same range in African ancestry. And Latinos are in the middle. And there's work recently from Matt Meyerson and Jian Carrot Zhang, who are at your neck of the woods, I think. They show that the ancestry is correlated with that among Latinos with EGFR mutation. And then they couldn't find a single locus, but they built a polygenic risk score for ancestry. And demonstrated the polygenic risk score for ancestry does better than any particular, than genome-wide ancestry. In other words, it's good just for genome-wide ancestry. And I'm just wondering, I don't know if you've thought about that kind of methodology, or what any thoughts about that as sort of a way to sort of say either we think that there's a genetic thing going on here, kind of need to dig more, or yeah.
Yeah, that's a really interesting, I need to read about that because it sounds really interesting. If you could send me the paper, I'd be keen to follow up. But as a population geneticist, I'm always like kind of nervous about potential hidden confounders. And I'm also, one direction my lab is going in is like trying to directly model things that we can measure from the environment, as well as genetic risk factors, and then partitioning how much of the phenotypic variation is explained by these covariates versus like genetic data. There's a really nice paper in Nature Genetics from, I think, Manny Rivas's group that did this for biomarkers, and we're able to show how much of the variation is explained by age, by sex, by a whole bunch of different, socioeconomic status, all sorts of different factors. And it varies a lot depending on phenotype. And so I know nothing about lung cancer, but I suspect that like this is going to vary a ton.
Yeah.
And I'd be interested to see how that would break down. Yeah, I'm keen to follow up. That sounds really interesting things.
Looking into that for what we're doing with the lab with HER2 in breast cancer. And actually, on that paper, they don't do out of sample replication of the local specific ancestry score. So that's kind of iffy based on what I'm doing with my own data. The second thing is I'm not sure they look at the correlation between the local ancestry score and overall and global ancestry.
Ancestry.
And they are likely to be highly correlated depending on the number of local specific ancestries they are averaging. So yeah, I think it was interesting, but I'm not sure.
More to be done methodologically. Okay, well, we're taking up a lot of people's time, and so I really want to thank you so much, Alicia, for being with us and telling us about your work. It's really interesting. I think it's really important, and I hope to have you back sometime soon, and maybe even meet in person.
Thank you so much. I really appreciate the invitation and always love a chance to meet with UCSF folks. Y'all are great. Thank you. Take care.